I'm going to talk about something I totally ignored yesterday, and that is how the messenger RNA finds the ribosome. Keep in mind, there are plenty of ribosomes in the cell. There are about 7,000 to 10,000 ribosomes per each bacterial cell. But there are thousands and thousands of messenger RNAs too. And so they're competing with each other to see who's going to be translated at the highest frequency. And that is all worked out evolutionarily over the years because there are certain proteins that are needed more than others. So the question really, though, is how does the messenger RNA find the ribosome, or how does the ribosome find the messenger RNA? And so the real question, which I posed here, is how do these two get together? What defines the site which is recognized by the ribosome binding site, or RBS? And this is a question that was key many, many years ago by Joan Stites at Yale, and Hindley and Staples in England at the time. And I just want to give you a view of what was known at the time. Everything was known to be able to do the experiment that they needed to do. In vitro translation assays were there. We knew the genetic code. We knew things started with AUG. We had messenger RNAs, but in those days, we didn't have messages coded by the genome, but rather phage RNA from viruses that infect bacteria. I had bacterial ribosomes essentially working with E. coli. Initiation factors, ATP, we knew was for charging tRNAs, GTP for the translation factors. And one key thing is Fred Sanger had worked out sequencing. So we could really ask specifically, what was the ribosome binding site? What was the sequence that was recognized by the ribosome? And of course, the experiment is essentially to take radioactive labeled messenger RNA, bind a ribosome to it, chew away the free RNA that wasn't protected by the ribosome, and then sequence that region and find out what it contained. What was the information in addition to the AUG, which was what they were looking for. Okay, so this is the experiment where they did bind the radioactive messenger RNA and then to the ribosomes, and then they put it onto a sucrose gradient, which is, as you know, a gradient of denser and denser sucrose. So the 70S ribosomes move furthest into the gradient 50S and 30S. And here are the 70S, and this is a radioactive peak of the message and the 70S ribosomes. This was taken from the gradient, and the sample was pelleted, and then they resuspended the pellet. And as I said, they had this ribosome protecting the message with a lot of messenger RNA on the side. So they treated it briefly with ribonuclease, and the only thing that was protected was the mRNA within the ribosome. And then they separated the ribosomes from the other fragments of the message by putting it through Cephidex. I don't know how many of you have used Cephidex, but it's a molecular sieve. It's like a ping pong ball with a lot of holes, so small particles get caught up in the ball, and the ribosomes move right through. And here you see the ribosomes moving right through, and everything else comes through later in the fraction. And then they sequenced what they had here. And the way they sequenced in those days, there's some actual people in this room who are still alive who used to do this. And he's in the back row. And you can treat the RNA fragment with enzymes that will cut at specific sites. A ribonuclease T1 cuts after single-stranded G residues, as they did in the top here, so it cuts after that G, it cuts after that G, this G, and so forth. And you get 4 or 5 long fragments and some short ones here. You line them up. You don't know how they're going to be arranged. But then you treat the same RNA fragment with ribonuclease A, and it cuts after U's and C's. And I think it's C's, yeah. And then you try to match up the fragments. And this is how, in those days, they were able to arrange the nucleotide sequence. Of course, they then had to take those fragments and know what the composition was. But the bottom line is that they detected a number of ribosome binding sites from different phage RNAs. And they lined up all the AUG sequences. And they had all this information. This is upstream of the start site, and this is the coding region of these messages. And the story is, and I don't know if it's true, but I think they took these data and they showed them to Francis Crick. And they said, Francis, what's the answer? What's the sequence that is recognized by the ribosome? And he was not able to detect what was in there that was really determining what the ribosome recognized. Shortly thereafter, 1972, 1973, two Australians revisited looking at the 3' end of 16S RNA. The 3' end of 16S RNA in the smaller subunit had been implicated by a number of studies with antibiotics and so forth to be the site of initiation. And it had originally been sequenced incorrectly, which sort of sidelined people for a few years. But John Shine was the graduate student in Dahlberg's lab, and the story goes that Lynn was on vacation, and John called him, and he came right back when he told him that he had just sequenced the 3' end, and he got a sequence that had a lot of pyrimidines, CCU, CCUUA. And they were actually excited because they were not only looking at initiation, but they were looking at termination. And the idea at that time was termination could be controlled by the ribosome by base pairing the termination codon. And if you look at the UUA sequence, that would actually base pair to UAA. And so the first half of their paper, where they report this new sequence, they're talking about the role of ribosomal RNA in recognizing termination codons, which was not correct. But the second half of their paper, they talk about the fact you've got a CCU-CC, and the fact you have a UU at the end there too. A pyrimidine tract, which later was shown not to exist in eukaryotic cells. So right where you have this bar here is where you have the CCU-CC sequence. So it was only in prokaryotes, it was not in eukaryotes. And the idea was this probably could interact with the polypurine-rich tract, which they found upstream of the Shinedalgarno, of the AUG. And so this sequence now is called the Shinedalgarno sequence, which I'm sure you're all aware of. And they showed in this paper the base pairing with different genes from these different messenger RNAs. Okay, that's all in theory, because they really hadn't shown that this base paired interaction occurs. So what Joan Stites did was to reconstruct this initiation complex using a messenger RNA that had 7 base pairs with the anti-Shinedalgarno region at the 3' end of 16S RNA. So this is the 3' end of 16S RNA. There's the 3' OH. And what they did was to actually form this complex with the message and then cut it with an enzyme that's made by a bacterium to kill other bacteria. It's called colicin E3. It's an enzyme that goes in and it makes a cleavage 49 base pairs in from the 3' end. You can imagine how lethal that is to ribosomes, but it's also very effective as an enzyme, which allowed Joan to isolate a complex of the 3' end 49 nucleotides and this piece of messenger RNA that they used to form the initiation complex. And then what Joan did was to, and I think it was Karen Jakes, they then purified this and they sequenced and they showed indeed there was base pairing, and so the Shinedalgarno model was now accepted. But the question really remained, was that all it was? Was it just the Shinedalgarno sequence that determined the affinity between a messenger RNA and the ribosome? I think a very interesting case is the R17 virus, which codes for 3 different proteins. They're all equidistant from the AUG site. These are the Shinedalgarno sequences for the A protein, the coat protein, and the replicase. But you notice they have different amounts of base pairing, 3 and 4 and about 7 here. So you would expect the one that only has 3 to be the one that's translated the least, and the one with 7 to be translated the most. They did 2 experiments. One, they added the intact virus to a translation system to see what was made, what were the ratios of the protein. They had those data also from in the cell, but the other was they translated fragments, which were essentially each of these parts of the total message separately from the others. And the translation level of the RNA fragments of each one gave what they predicted. They got 40, a ratio of 40 to 1 to 4, as you can see here. However, if they translated the intact messenger RNA, the story was much different. There they got 20 for this one that has the least amount of base pairing with the Shinedalgarno, 1 here which has the most, and 5 for this. So it was completely reversed. So that indicated that it was more than just the Shinedalgarno that was important. So the question now is, what are the other regions to look at, and how might they each contribute? So we have the upstream region of the message, the 5' end, then the Shinedalgarno, and then what's called the spacer region, and then the AUG, and then this is the coding sequence for that particular gene. So what I'll do now is just go through each of those regions and show you some data to suggest, and the bottom line is that each one of these regions contributes to the affinity of the message for the ribosome. And before we even get there, let me just point out that AUG in itself is not sacred in the sense that you don't need to have AUG as a start codon. You can actually have UUG, GUG, and other sequences here too. This was work done by Michael O'Connor in my lab a few years ago, and he took all these different messages, changed only in the AUG, the initiation codon, and you can see the differences in level of translation, 25, 7, 11,000. And when you get down here, finally this is a stop codon, so you essentially get nothing here. But you can even have initiation from sequences like AUC or CUG. What Michael did also was to take the initiation factor mutants he had in his freezer and show that if you had mutant initiation factor 3s, they would actually affect the level of recognition of these different messages such that now some of these more unusual initiation start sites were enhanced, say in here, 7 times or 6 times what they were when they just have normal IF3. But this again shows that the regulation of translation is not only, again, the information in the messenger RNA, but also the factors that recognize the messenger RNA. Okay, so the other thing was that people began to look at lining up all the AUGs for these messages. They recognized right away that the regions that base paired with the 16S RNA were both variable in the number of base pairs that were complementary to the 16S RNA and variable in the distance from the start site, the AUG. So that called for an experiment where you would vary each of those two and see what you get. So this is done by Larry Gold. A lot of this work is done by Larry Gold in Colorado. And what he did was to take 2 different messages, and one had more complementarity to the 16S RNA Shinedalgarno, and another one had less. So this one had 5 base pairs, and this one had 8 base pairs in the Shinedalgarno sequence complementarity. And he looked for the level of translation of these messages when he would have varying amounts of A residues between the Shinedalgarno sequence and the AUG start site. And as you can see, if you only have 2 or 3 A residues between the AUG and the Shinedalgarno, you essentially get no translation. And we didn't know what that meant initially, but as we know now the crystal structure of the ribosome, we know where the AUG is, in other words, where the P site is in the ribosome, and we know where the 16S3' end, it's the anti-Shinedalgarno region, and the distance between the P site and where the Shinedalgarno region is such that you need at least 3 bases to span that, because it can't have the P site occupied with the AUG and reach over into that region. So the more spacing you had between the AUG and Shinedalgarno, the better the translation. But eventually you get so that there's so much space between the AUG and the Shinedalgarno that it's getting in the way, and it's sort of going into that interface between the two subunits, and you begin to inhibit translation. In fact, it loops into the area where the E-site tRNA is trying to occupy. And then the less Shinedalgarno complementarity gave you less transcript. Okay, so the distance and the degree of base pairing was important. Then Larry started looking, actually this is done by Anna Hu, and this is done in the Netherlands. And this is work where they began to ask the question, what's the importance of the region between the Shinedalgarno and the AUG? And they looked at the sequence just before the AUG, okay? They're saying, well, that's not terribly important. That's upstream of the start site. But it's in the region between the AUG and the Shinedalgarno region. So it's really the equivalent of the E site of the messenger RNA. But it's not a piece that's translated at initiation. And so they constructed all kinds of codons. The only thing they changed was the equivalent of the E site in these messages. And they asked, what's the level of translation? And the relative level varied from 0.1 up to 2.2. So if you had the E site with a UUC, it would be 0.1 relative to the normal translation rate. And if instead of UUC, you had a CUU, it was 22 times more effective in translating. That was really quite interesting because this is a region that's not translated. It's not got a tRNA going in, and yet it was 22 times better. Then they asked the other question. This is Larry Gold's work, and no, this is also Herman de Boer, sorry, and Piet van Nippenburg. And what they did, again in Holland, and what they did was now to look at the other side of the AUG. Instead of the E site, they looked at the A site on the other side of AUG. And again, they found sequences where it was 0.1, and then they found sequences where it was 1.5. So you have a 15-fold difference here. You have a 22-fold difference on the other side of the AUG. I don't know if they ever put those two together to see if they would get a 300-fold amplification of translation. But what they really did was to show the significance of the nucleotides on either side of the start site. And these are called the spacer region or the coding region. Another point which relates to the earlier story of the relative translation of those three different genes was done by Jan van Douwen, again from the Netherlands. And he took a message that has a lot of base pairing around the Shinedalgarno region and the AUG start site. And then this is the coding region here. And if he changed this AU base pair to a GC, he dropped the translation rate down to 4% what it was. And if he changes UU into an AU, he dropped the translation rate down to 1,000th of what it normally is, and so forth. So you can make secondary structures so tight that you can actually make it impossible to translate these. But this really wasn't part of the story that related to specific sequences, but it showed the importance of secondary structure. So as people read more and more about other people's works on the importance of the E site or the A site or secondary structure, they began to look all over the messenger RNA. And Irina Boni in Russia actually found sequences that are tracks of poly-U upstream of messenger RNAs. And she found that these also enhanced the translation of different messenger RNAs. The explanation here probably is the fact that there's a protein, the largest protein on the ribosome called protein S1 on the small subunit has a very high affinity for polypyrimidine tracks. And this is the explanation that we think accounts for this enhancer element. There were other people who thought, as they did a lot of looking at messages, and they thought, okay, here's an interesting sequence that seems to be translated more than it should. And they were looking for complementary sequences between the ribosomal RNA and certain messages. And Ernst Fuchs found a number of messages he thought were actually complementary to the right side of helix 44, which we talked about yesterday, which is a region involved in decoding. And his model was that helix 44 actually came apart and the message base paired to the sequence on the right side of the strand. Well, this is a highly theoretical and very difficult to prove, except that Michael O'Connor, again, a fellow in my lab, thought of an experiment where he took the ribosomal RNAs like this, of this helix 44 stem, and he reversed them. And when you do that, instead of having the sequence go 5' to 3', now it's over here going 3' to 5'. So it's completely different. But the structure, the overall structure is the same because the base pairs are all conserved. These were viable ribosomes, and so he then assayed these ribosomes, which he called the mutant anti-downstream ribosomes, having this reverse, to see if they affected the level of translation, because now they couldn't complement the sequence that Fuchs was saying base paired, because the base pairing was now parallel instead of anti-parallel. And so he looked at these different messages and looked for their level of translation in wild-type ribosomes and ribosomes where he had reversed it, and what he found was they were exactly the same. So at least in this case, the downstream box sequence was shown to be not correct. It's interesting because the proposal that someone made here looked sufficiently convincing that it actually took a lot of work to disprove it. And that's just as important in science as proving things, because otherwise, if things don't get answered, sometimes they never get answered. It's not that you have a grudge on someone, you just want to find out what the truth is. And Ernst is a really nice fellow, but I think those data are fairly convincing. So what are the elements of the ribosome binding site? AUG, or GUG, or UUG. Complementarity of the Shinedalgarno region, the length of spacing between the Shinedalgarno and the AUG, the sequence of the spacer, meaning the E site sequences of 5' of the Shinedalgarno, which is a poly-U tract, and 3' of AUG, which is the A site, secondary structure, and enhancer sequences. Okay, so what is the ideal ribosome? What's the optimum ribosome binding site? It really depends on how much protein you want to make from that message. So an optimum for one protein will be not really good binding to the ribosome, and another one will have a high affinity if you want the protein to be produced. So nature has made a careful selection in these bases for years and years and years, selecting just the right amount of protein, keeping in mind that in bacteria, the regulation is not as refined and as regulated as it is in eukaryotes. There, it's a little more difficult to do these kinds of experiments. Phenotypic effects of mutations in the Shinedalgarno and the anti-Shinedalgarno region. What about those? So just think about it. If you take a messenger RNA and you make a single base mutation in the region that is the Shinedalgarno region, you can actually step up or step down the level of translation. Okay? That's pretty obvious. What's the effect, however, if you make a mutation in the 16S RNA in the sequence that base pairs to the Shinedalgarno sequence? Can anyone? Okay. I always hated it when they asked me in class whether I would volunteer. Does anyone want to predict what would happen if you change a sequence in the ribosomal RNA anti-Shinedalgarno region? What's that? Right, so if you change a sequence like in the CCUCC sequence, what we did in our lab was to change the third C. So instead of CCUCC, we made it CCUUC. We figured going from one pyrimidine to another is going to be okay. And it worked in the sense that all of where the complementarity went down, we could see less of each of these proteins in a two-dimensional gel. And where the complementarity went up, the level of translation went up. But the main point is, when we introduce this mutation into the cell, it killed the cell. Because it's taken eons for the cell to evolve the right amount of each of these proteins in the cell. And all of a sudden, we've shifted it. And it just doesn't make any sense to the cell. So the cell can't survive. OK, and Herman de Boer has made what are called specialized ribosomes, where he's changed the anti-Shinedalgarno region of 16S and made it, instead of CCUCC, he makes it GGA-GG. And he makes some messages that complement that. So these specialized ribosomes will actually translate only specialized messenger RNAs in the cell. OK, now I'm just going to briefly talk a little about where does a message go when it does find the ribosome. And there was a group in Russia. This is a model by Vasilia of the 30S subunit, which doesn't have the appearance of a lot of structure, but it turns out to look exactly like what you see in the crystal structure. So here's the head, and this is the shoulder where the messenger RNA comes around. Ivan Shatsky actually was a person who proposed this model with the message going around here like this. And when the crystal structure was determined for the message on the 30S subunit, you can see the message going in here, this yellow and red, going around and then coming out here, just like it did right there. And over here, the message, now we've turned a little. You can see the message coming around the side here, just like that. So the structure they had worked out before crystallization turned out to be a beautiful predictor and very complementary to what we saw in the structure here. This just gives you a cryo-EM picture, and to show you that what you can't see so well in the crystal structure is that part of the RNA looks like it loops over and forms a channel for the messenger RNA, the white line here, to go through the message around the neck and out that way. And of course, that's exactly where you would have the 3 tRNAs base pairing to the message as they decode the message right here. And this is the way they fit into the 50S subunit. And once again, looking at the message as it goes into the ribosome, you notice there are 3 proteins here, S3, S4, and S5. We talked about S4 and S5 yesterday, which were involved in the open, closed. They were on the back, and this is the back. The decoding is going on the other side over here. The interesting thing about these 3 proteins is it looks like they have some kind of a helicase activity. At least they're able to open up the secondary structure of the ribosome. They have a number of arginine residues, which will interact with the sugar phosphate backbone of the message and form an interaction sufficient that it holds the message on the outside here. And if there is a secondary structure, not too strong a secondary structure, as the ribosome is decoding and going like this, they are holding the message here, and you stretch the secondary structure to allow the message then to move in through here. So we think those 3 proteins, Harry Nowler actually changed some of those arginine residues and found that it affected the ability to open up secondary structure. So these are important with that function as well as what we talked about yesterday. Okay, this is now looking at the message as it's coming into the decoding region, the P site, the A site, and this is the E site. And then up here is upstream with the Shinedalgarno sequence, which is base pairing to this part right here, which is the anti-Shinedalgarno sequence of the 3' end of 16S RNA. So we know something about specific regions of the ribosomal RNA. And you know that we talked yesterday about 1492 and 1493 flipping in here. This is just to give you an orientation of where they are relative to the message and the picture of the message moving through here. I told Galko I would finish quickly, so what I want to show now is the tRNAs actually base pairing to that message, and taking those tRNAs away from that decoding region, we can now say this is the path of the messenger RNA through the ribosome. And you know, there's a tendency when you have structure to say, this is it, this is the answer, this is all I really need to know. I know exactly where the message goes in these ribosomes. I know exactly the path it's taking. If you just knew the structure here, and you said, look, this is exactly where it goes. Look how that's just fitting right through there. If you didn't know the history of the study of ribosomes and message, you wouldn't realize that up here in the 5' end or the 3' end are sequences that will determine whether that message will be translated 300 times better than another message where you have a different A site and E site around the AUG. So this is where I think it's important to keep in mind that you have to know not only structure, but you have to know function. And I think a lot of the work Larry Gold and Herman de Boer have done over the years has been not neglected, but no one has gone back and constructed some of those extreme mutants in the A site and the E site to see how they actually interact with different regions of the ribosomal RNA to account for the fact you can have this enormous difference in translation rate depending on just those few base modifications at those sites. I don't know who would want it. That's an enormous amount of work. Well, it's not that much work, but it involves making the message and soaking the messenger RNAs into crystal structures. But we do know some things about that. For example, there is a base called 926, which has a phosphate, which is interacting with a phosphate upstream of the AUG. We know that's important. So, I mean, we already have some clues that if we did that sort of work, that we would actually see at a molecular level exactly why we have these enormous differences in the translation rate of the messenger RNA. Okay, and that's, I think I did it.